Hello, everybody, and welcome to this week's Imaging One World talk. Just as a quick notice, there is no talk next week because it's the Microscopy Microanalysis Congress is on in the UK. We're being hosted by RMS in the UK, so we'll have a break for a week, and then it's back to talks as normal after that. It's with great pleasure that I can introduce Shan, who's our speaker this week. So thank you very much for agreeing to talk to us. Shan is a senior scientist and associate director of FIB-SEM technology at Janelia Farm in the US. So, and really, this is the institute that has really pioneered the revolution in FIB-SEM that, well, I guess you'll talk about today. Shan has a really long history in technology development, going back into the sort of early 2000s. 2009, I think you joined at HHMI at Janelia and really started working on FIB-SEM, and now you're sort of basically leading the work there. And I think in your background, you mentioned that's a sort of a shot of the current facility, which has about sort of 8 FIB-SEMs running 24/7 behind you, plus a few more round and about the building. I think you've developed FIB-SEM from the sort of first generation of just high volume imaging through to the second generation with a really advanced high throughput added to all of this, and now I think you're on sort of FIB-SEM 3.0, which is artifact-free 3D imaging. So I'm really interested to see what you've got to say about that as well, because this is just a field that continues to develop with pace. I guess what I should do is just let you get on. Thank you very much. So over to you, Shan.
All right, thank you so much, Nick, for the invitation, a very kind introduction. Let me share the screen now. Can you see the screen?
Yes.
You can see the great screen, yeah, thanks.
All right, so let's see here. It's a great pleasure to meet everyone virtually. I hope we'll meet sometime in person. Over the past few months, if you were and listen to this series, you must have heard FIB-SEM related talks from my colleagues, Harold Haas, Jennifer Lippincott-Schwartz, and Aubrey Weigel. This is probably the final talk about FIB-SEM from Janelia, I think at least for a while. So there might be some material overlap. I apologize if you've seen some of them before. But today I want to present this enhanced FIB-SEM from a slightly different angle, focusing on the instrumentation development as well as the capability side of the story. So of course, with EM, we're not looking at live samples anymore, but I hope these frozen in time snapshot will provide a new perspective on how we visualize the cellular world at nanometer resolutions and in the context of whole cells and tissues. So these are some examples from our studies: a brain circuit of a fruit fly, a mouse pancreatic islet beta cell, and a piece of mouse liver tissue. So this work is I'm presenting today is a massive teamwork. So I want to first acknowledge many of colleagues from Janelia, particularly the FIB-SEM technology group, as well as the Haas Lab. Also our wonderful external collaborators. They are the ones that bringing these interesting questions I'm going to share with you today. So let me start with a quote from Sidney Brenner, a Nobel Prize laureate of physiology in 2002. Basically what he said was, progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. So let's talk about some new techniques first. So I hope in the next hour or so, I will take you on a journey of how to achieve more than 4 orders of magnitude improvement in imaging volume using focused ion beam SDM. thereby enabling a wide range of discoveries from single cell to tissue biology, such as connectomics that contains more than thousands of cells. So what motivated us a decade ago was this grand challenge. I want to obtain a complete circuit diagram of a fruit fly. So complete means every single neurons and every single synapse needs to be identified. So before 2020, I would say there was only one complete wiring diagram exists for a nematode called C. elegans, which has exactly 302 neurons. That wiring diagram took Sidney Brenner and his team 10 years to reconstruct. So the fruit fly has a total of 100,000 neurons, so much bigger. And given its volume and the required resolution, so it's almost like taking, depends on where I go, give a talk, I will use an analogy differently. It's like taking a 3D picture of the Big Ben Tower at the resolution of a sesame seed. So it's quite a daunting task. However, the most challenging part is not about the size of the data or even how long it takes, even though it's years, but how perfect it needs to be. So basically, we cannot afford any gaps more than 100 nanometers, which poses a substantial challenge to trace these neurons across that gap. So let me just use brain circuit reconstruction as an example to illustrate the process flow of volume EM microscopy. So we start with a piece of a fresh tissue. Unfortunately, it's not compatible with electron microscopy. So first thing it needs to do is fix the tissue using either chemical or high pressure frozen, this low temperature. Basically what it does is to lock these biomolecules like proteins or lipids in space. Then we go through the what we call a heavy metal staining that provides contrast to the electron microscope. And a couple steps like a dehydration and resin embedding. So at the end, you have a piece of plastic with heavy metal inside. Just keep in mind, what you're imaging with this piece of sample is not those biomolecules you want to image. It's just the heavy metal you put in that located at where the biomolecules used to be. So once you have a sample, there are a variety of methods you can choose to image such volume. For example, you can use a sharp diamond knife to cut this piece into thin sections, about 40 to 50 nanometer thick. And then you can image that section using either TEM, transmission electron microscopy, or just lay them out on a piece of a silicon wafer and image them through SCM, scanning electron microscopy. So the Z thickness pretty much determine the Z resolution. Now, you can also go with a different approach, what we call a block-based approach, pioneered by Winfried Denk. You first look at the top surface of this block using an SCM, and then just shave off a thin layer using, again, a diamond knife. Now, because you no longer need to maintain the integrity of these thin sections, you could actually go much thinner for the Z. So you just repeat the imaging and shaving process over and over until you get the entire volume. So even though we reduce the Z thickness down to 20 to 25 nanometers, it's still an order of magnitude worse than the XY resolution electron microscopy can provide. So you can switch out the diamond knife, use the focus IMB. So now all of a sudden you get rid of any mechanical movements using high energy ion to blaze off just a few nanometers of material. In fact, you can get down to as thin as like 1 nanometer along Z direction. But it's still the same process. You go through the milling imaging process and just repeat that cycle tens of thousands of times until you have the entire volume image. So it doesn't matter which method you use. At the end, you're going to end up with tons of images. You first need to stitch them together in plane, and then align them along the Z-axis to form the 3D volume. Now, for brain circuit reconstruction, the next step is basically using computer algorithm to segment out this volume to say, okay, assign pixels to specific neurons or synapse. But that process is not 100% error-free. So at the end, we need human proofreaders to proofread that process, the volume, the result, basically correct any mistakes that the computers made. So so we choose FIB-SEM, and the reason is quite obvious, because FIB-SEM provides this isotropic voxels less than 10 nanometers, compared to, say, an isotropic voxel obtained from these diamond knife cut sections. They are like, say, 40 nanometer thick. So this is very critical if we want to follow neurons along all three directions for neuronal tracing. So you will have a little bit hard time here with the anisotropic resolution along XZ or YZ directions. But resolution is only part of the equation. So we also need to consider a volume. So this is the resolution volume space I always like to use to compare different modality of EM volume EM methods. So on the y-axis is the isotropic voxels, or the best isotropic voxel one technique can offer. On the x-axis is the size or volume. They're both in log scales. So you see this wide open space right here, not covered by any of the existing technology. But I want to argue this is probably most of biologists are interested in, because it offers, say, less than 10 nanometer resolution, volume as large as like 1 millimeter cube. These conventional FIB-SEMs are great in offering resolution less than 10 nanometers isotropic, but the volume is rather small. It's about the size of an yeast cell, not quite an entire mammalian cell if you want to do at 8 nanometers or less. And the fibrane, as I mentioned earlier, it has a volume about 40 million cubic microns. And you can see there's a huge gap, more than 4 orders of magnitude, big gap right here. So how do we bridge such a gap so that we can use FIB-SEM to image fibrane? And before I talk about how, let's look at why such a gap exists at the first place. Say you have $1 million spent buying a brand new FIB-SEM and put in a sample and turn it on, and this is typically what you get. So here is a culture cell sample. You can see the nucleus, and here's another cell, also nucleus. But after a few hours, say less than 4 hours, you realize, oops, your image lose focus. So what do? So you pause the machine, do some manual focus, or use some routine provided by the software, and focus the image again, say at 5 hours. But then it doesn't last long. Half a day later, same thing happened again. So you lose focus, and then you go through the refocus exercise. So within just 24 hours, you already have a couple of these episodes. And to image the entire fibrane, we need years of operation. So obviously, there's a big problem here. So first of all, we have a system drift that only lasts for sometimes in the orders of hours. And then there are a long list of interrupts ranging from days to years. The most challenging part actually is after any of these interrupts, how to restart image acquisition without losing data. Because we are talking about not only nanometer resolution in the imaging side, but also in the milling side. So FIB-SEM, as you are aware by now, is a destructive and forgiving process. So any of these interrupts, pretty much we couldn't restart it easily. So that's the end of the run. So let me just draw you to an interrupt that called FIB source reheat. The ones who are familiar with it, you know, every 3 to 5 days, you need to reheat the source, just like you need to refill your gas tank. And that had to happen every 3 to 5 days. So combined with slow imaging rate, because SCM, you're doing pixel by pixel rather than like TEM, a parallel process, this end result is you will have small volumes out of these commercial systems. So how did we solve this problem? Now, of course, you can try really hard to improve the reliability to 100%. or you can tackle this issue from a total different angle. So long story short, I redesigned the entire control system and developed what I call a smart architect that's immune to interrupts. My philosophy was to create a smart system with 100% effective reliability that's not afraid of interrupts, and most importantly, is capable to recover from these interrupts and resume imaging without defects in the final image stack. Now, besides this major architect change, there's a long list of packages or features, which I won't have time to go into much detail, but just highlight a few of them here. So if you look at a FIB-SEM system, zoom into looking inside the chamber, this is what the configuration is. So you have the SCM coming from the top, taking the picture of the sample, and the FIB is coming at an angle relative to SCM, typically 50 to 60 degrees. Because the mechanical constraint between these two columns, the sample has to be a little bit further away from the SCM column. And it's at an angle after the FIB surface is milled off. So this is really doesn't quite make sense when I first look at this. It's just like an analogy I always like to use is if you want to read a book, why do you want to have a book at an angle and far away from you, right? So what makes sense is you want to steer the book straight on and move it closer to your eyes. Now, luckily, it's not too difficult to solve such a problem. All you need to do is just reposition the FIB column to be 90 degrees relative to the SCM column. So now all of a sudden, the mill surface is perpendicular to the SCM beam, and you don't have a mechanical constraint. You can move the sample a lot closer to the SCM column. So with such a 90-degree configuration, immediately you get better and stronger signal. So you can image faster. Now, a second very important feature we implemented is it's a closed loop FIB milling. Because FIB is destructive in this case, we monitor 3 different currents, one from the specimen and 2 from 2 different beam dumps, and use that as a feedback mechanism to monitor where the FIB is actually down to nanometer accuracy. So this is critical to ensure precise milling during normal operation, and most importantly, allows smooth re-engaging after any of those interrupts without damaging the sample, and then we can go straight into resuming the FIB-SEM process. Now, recall the system drift I showed you earlier in the movie. I also create this, what I call a inline image optimization routine with no overheads. Basically, we'll keep the SCM image in focus, not just within hours, but days, weeks, to months. So combine all of these together. At the end, what you get it's a very fast system, and that can continue to run months to years. So with such capability, I think we're ready to look at some exciting discoveries. Coming back to the fly brain, we haven't got the entire brain yet. So this was a sample we collected a few years ago. We call a hemibrain. Now, the reason why we choose this area because it is really the control center of the fly. So even though it has less than half of the volume, but the function is more than half. So it contains the navigation as well as the learning, olfactory, and visual system of the fly. So it took us 2 years on and off with 2 machines. Now, the way how we handle this is first we partition this big volume into smaller pieces using the hot knife developed by Ken Hayworth, my colleagues. Each section is about 20 micron thick. And then you can image these tabs in parallel in 2 machines. So as you can see, the system is very heavily customized. Here, I mounted a FEI, now thermal Fisher, focus ion beam column onto a Zeiss column. So conveniently, I named that FIBSEM. So after 2 years on and off on those 2 machines, we have a total 13 tabs. The volume is about 16 million cubic microns. Let me just play the movie. I remind you the task, what we want to do is to trace every single neurons and identify every single new synapse. Of course, the first question you will ask, these digitally restitching column sections, are we able to trace the process through? So what you're seeing here is one of those interface that we hot knife earlier and then digitally restitch the image back. And the goal is to trace neurons from one section going into the next one. The answer is not too bad. So actually we could follow pretty much everything through that the interface. So check. Another thing I want to remind you is within that 2 years on and off 2 machines, there are literally more than 100 FIB repeats. So a lot of interrupts, but you don't see visible gaps right here. So that's what I meant 100% effective reliability. So we have all these volume line stitch together, then we send it to Google. A couple of weeks later, this is what we get back. And this is what we call a segmented volume. Basically, they use this state-of-the-art flood filling network AI network to identify which voxel correspond to which neurons. So different color represents different neurons. And of course, the algorithm is not 100% perfect. So there are some challenging areas showing in the lower right here that require corrections. So with the help of 50 proofreaders working continuously for 2 years to correct those mistakes of the automatic segmented machine learning, we now have the largest connectome today with more than 25,000 neurons and 4,000 cell types. So just put the volume into perspective. The neuron, so the hemibrain size is about 16 million qubit microns, and a commercially available FIB-SEM can typically do about 1,000 qubit microns 10 years ago, and unfortunately still about the same today. So the bottom line here is that with better imaging quality, segmentation, and proofreading tools, we achieve a couple orders of magnitude improvement in the connectome pipeline throughput, not just imaging, it's the pipeline, compared to where we when we started 10 years ago. So we think this data is very valuable, so we put it into a public accessible website called Newprint. And over there, you can browse the entire raw data in SCM as well as the segmented in the 3D fashion. And here's the connection matrix between any 2 neurons. Now, in addition to neurobiologists, this actually has a lot of useful information. Say if you're a cell biologist, think about it, you have 25,000 cells. So I'll just give you an example here. I was browsing this data set one day, and then I found this beautiful, actually the ER sheet in one of the cell body area, and then just zoom in and just look amazing. So with this rich cell biology information, I'm sure someone could find some interesting discovery in addition to the neuronal circuit connections. Another example is mitochondria. In fact, there are more than 6 million mitochondria in this hemibrain volume. And we immediately notice there are kind of 3 kinds of mitochondria. One we just call dark, and then the light on the other end, and then somewhere in the middle called medium. So it'd be good that we can identify these 6 million mitochondria in the volume. So the approach we took is pretty much identical to the wiring diagram. We first have a high resolution volume that train a neural network to recognize these different classes, and then just apply that network into the entire volume. So now all of a sudden we can quickly identify the classes of the category of the mitochondria in this entire volume. You can see maybe that relate to certain energy consumption or something probably very, very interesting to explore further. So coming back to this operating space, so what we have done literally is just create an enhanced version of the FIB-SEM, addressing the limitation of a conventional system and substantially expanding its capability. So the entire Drosophila brain is within reach. As a matter of fact, these 8 machines behind me have been running 24/7 since early this year. So for the entire central nerve system of the fly, we target to finish this whole project by end of this year. So with this newly enabled capability, biologists have much greater freedom to choose their operating conditions based on their applications. So oftentimes with fixed amount of time, there's always a trade-off between resolution and volume that's indicated by these red dashed lines here. So you can, for example, if you can live with bigger voxels, you can go with the high throughput mode, or you zoom into a smaller volume using high resolution to reveal finer details. With the remaining time, I will show you a few examples that demonstrate the unique capability offered by this newly available regime from enhanced FIB-SEM. So coming back to the wiring diagram, I think the ultimate goal of obtaining a wiring diagram is to link behavior to neural circuits. As we know, behavior studies are intrinsically noisy and require a lot of statistics. So it's very helpful that our collaborators, Albert, Marta, and Philip, picked the larvae of Drosophila. It's about 10 times smaller in volume compared to the adults. And we choose high throughput mode with 12 nanometer voxels. So instead of years, only a few weeks are needed to obtain the entire wiring diagram. So here's how the experiment works. So our collaborator first performed whole nerve system imaging at cellular resolution to capture brain activities of a specific behavior using light sheet microscopy. So each flash here indicates a neuron firing. We then image the exact same animal using FIB-SEM. Finally, the 2 volumes are overlapped, so we know which neurons are active within the complete circuitry for such behavior. So what FIB-SEM was able to help in this case is to quickly image multiple samples using high throughput mode while maintaining sufficient resolution to correlate back to the optical measurements for behavior. So at the end, we can answer questions such as which neuronal circuits contribute to a specific animal behavior. Next, this is a collaboration we have with Andreas and McKelly from Dresden. They are interested in, the pancreatic islet beta cells. So the main function of these beta cells are to synthesize and store insulin in the secretory granules showing in these little black dots here, and to release them under high glucose stimulation. So what is the structure basis to fulfill such a function, right? So Andreas is particularly interested in the role of microtubule network, which is still under debate. So one model you can imagine is they are just the transport tracks that pull these secretory granules to plasma membrane for exocytosis. Anyway, this is still an important subject for physiology study as well as diabetes research. So to answer this question, we first FIB-SEM image multiple whole beta cells, and Andreas and his other collaborators then segment out the entire volume, looking at various intracellular organelles, such as secretory granules showing in these orange balls, and there are 5 to 10,000 of these secretory granules in each of the beta cells. They are everywhere, basically. And now you can also look at the mitochondria in blue and Golgi apparatus in green. And finally, the microtubule network, the main subject of the study. And the data volume is large enough that encompasses multiple whole cells, as I said, for statistical analysis. Indeed, we have 4 high glucose conditions and 3 low glucose conditions. So after segmentation, a series of comprehensive and quantitative spatial analysis can be performed for all 7 beta cells. And here I'm just showing one of the examples that Andreas analyzed the length and curvature of microtubules. And as you can see, at low glucose condition, the microtubule is about 3 times longer than the high glucose condition. But there's not much change in terms of the curvature. So after a series of analysis, it comes to an overall finding that can be summarized in a simple cartoon form like this. So of course, first you see lengthy microtubules in the low glucose and fragmented in the high glucose. And also you notice there are a lot more secretory granules right next to the Golgi and under high glucose condition. So one explanation of how microtubule network regulate insulin release could be that the tightly connected microtubule network hinders the transport of secretory granules at low glucose condition. Upon high glucose stimulation, such a network loosens up and allow secretory granule to reach the plasma membrane easier for release. Now, the next example is about an organ right next to the pancreas. It's the liver. Our collaborators, Ganesh, Anna, and Gokhan from Harvard are interested in the liver tissue, and particularly the ER structure in these hepatocytes. So they hypothesized the function diversity of the hepatic ER is due to its structural regulation. They really want to understand the relationship, again, between function and structure. So in order to address this question in its native tissue environment and in the context of metabolism, they want to obtain a detailed 3D information of the ER structure and organization in liver tissue, which is very different than the culture cell under various physiological and pathological states. Again, we started by imaging multiple tissue samples of different conditions. So what I'm showing here is the lean tissue on the left, obese tissue on the right, and they are all large enough to contain multiple cells. And you can already see the morphological difference between the lean and the obese in the raw image stack, and which becomes a lot more obvious after segmentation. For example, you see a lot more lipid droplets in yellow here in the obese, which is not surprising at all. We can zoom in to take a closer look at the ER structure of these two cases. Now, all of a sudden, you see these beautiful ER sheets in the lean tissue, and then these sheets kind of turn into tubules in the obese animal. And as you can see, we're looking flying through one of the tubules here. Now, with segmentation, we can quantify such observation. For example, if you look at the sheet to tubule ratio, you see actually more than 50% reduction when you go from lean to obese animal. But the story didn't end here. Ganesh and Anna wanted to find out if the metabolism functions can be recovered after the structure is restored. So they use a protein called CLEM63, which can restore the ER structure back to the sheet-like, as you can see in the EM image, as well as a statistical analysis. Interesting enough, the metabolism function returns after the structure was recovered, which is really exciting because that could pave the path for potential therapeutic solutions for obesity down the road. So far, I talked about imagings at standard like 8 nanometers and 12 nanometers. It'll be very interesting to go into smaller volumes at higher resolutions. This is one of the examples I'm showing here. It's a whole entire HeLa cell that image at 4 nanometers voxels. So it's substantially more challenging at 4 nanometers in terms of system stability and reliability. But the good news is that the rich information offered by these high resolution data sets is just incredible. I hope all of you have found your favorite organelles. Potentially these can be segmented out or classified in the entire volume. For example, you see the centrosome here, you see the microtubule with a hollow core, as well as a polyribosome chain. But the bad news is it took almost a month to acquire. So then I focused my effort to further improve the stability and signal detection for faster imaging at this high resolution. So the next movie basically highlights the results of such effort and the value of 4 nanometer resolution versus 8 or higher resolution, larger voxel size. So what you are seeing here on the left is the 4 nanometer sampling. On the right, it's the emulated 8 nanometers. So when you look at, say, this close contact between organelles like ER and mito, it really helpful to have higher resolution so you can see the contact, as well as, say, microtubule, you see the hollow cores at 4, but just like a fuzzy stick at 8 nanometers. So this took, we see the value, but the volume was somewhat limited before. Say we can only get about 500 microns, cubic microns in 5 days. Now with better configuration of the machine and stronger staining, we actually were able to achieve more than 200 times bigger volume. So as you can see, it's much easier to study these close contacts between various organelles with higher resolution. So to obtain these high resolution whole cell data require a considerable amount of investment in technology development, equipment resources, and imaging. But the resulting wealth of data is just invaluable and comprehensive. So knowing the value of these data sets, so we create a reference library of a dozen wild type samples from cancer cells to immune cells, and from pancreatic islets to neural tissues. And we provide open access of these high resolution data sets that we have generated over the years via Open Organelle website. We hope this resource could add value to biologists. In particular, the examination of a wild type cell can encourage further experiments to compare, say, features against perturbation of disease, mutations, development environments at the whole cell level. So let me show you a T-cell attacking cancer cell example from that reference library. And this is a collaboration with Alex and Ira from Genentech. As we know, T-cells are immune cells that have the capability to identify and destroy virus-infected or cancer cells. Upon encountering the target, T-cell will latch onto the target cell to form what they call an immunological synapse, and then punch through the plasma membrane of the cancer cell and inject a chemical weapon to kill the cancer cell. The entire process happened in a matter of minutes. So it'd be cool to capture the moment of such an attack. So let's take a look at this using FIB-SEM. Here showing a T cell in red, cancer cell in white. So first, let's look at the raw image data, the FIB-SEM black and white images, and you can see here's the cancer cell has a T cell. So it'd be a lot more helpful if we can know which volume corresponds to what. That's why we do a quick segmentation to separate the T cell volume from the cancer cell volume, so that you know where the synapse is. So cancer cell here is in blue, T cell in brown, and this non-labeled area is the immunological synapse. Basically, the battleground, the front line of the attack that we're interested in. So as you can imagine, when the T cell punch through the plasma membrane of a cancer cell have this self-defense mechanism. So really want to push out the things that get injected in. So you can see these aggressive protrusion coming back out from the cancer cell at the 3D volume here. So what I'm showing here now is another classic signature of a T cell attacking cancer, the immunological synapse, what they call cupping. So you see this basically forming a big jar. So what T cell is trying to do is to maximize the surface contact so that you have the best chance of an attack. Here's another example. The centrosomes are polarized towards the cancer cell, and you see this purple lytic granules. These are the chemical weapons that T cell use to kill the cancer cell. Now, of course, in addition to segment out the T cell, you can segment the organelles inside the cancer cell. It's just showing this microtubule mitochondria network. So this is the first time we capture this comprehensive 3D picture of an attack and anti-attack event. So all the electron microscope I've shown you so far is black and white, right? So it's always challenging if I ask you to identify, to tell apart these three apples with just a black and white picture. And that's where we need help from some photons. And that's what we call the correlated light and electron microscopy, or CLEM. So with color, it's much easier task that we can set apart these three apples. Back to the EM image of biological samples, we see all kinds of intracellular vesicles from similar size and shape. So are they lysosomes or peroxisomes? it's often quite challenging to distinguish them with just the black and white image. However, if we fluorescently label a specific peroxisome targeting sequence, in this case, we can immediately know which ones are peroxisomes. In fact, peroxisomes have all different shape, and this is just like a ball, and here's like a flat pancake, and this is almost like a donut. Likewise, we can label other organelles to study the close contact between them. So the first example shows a close contact between a peroxisome and endosome. And second example also shows a close contact between endosome and peroxisome, but just in this case, the endosome, as you can see, has a very different shape compared to the first one. It's like a very long tube rather than just like somewhat like a blob. The last example is quite interesting. It's the peroxisome have a close contact with ER sheet that wraps around the one mitochondria. So let me show you a movie, a beautiful one that created by David Hoffman that contains 2 COS7 cells with this cryo-clamp approach. So first we look at the 3D rendering of the FIB-SEM. This is just a pseudo color, not fluorescence, just to clarify. The orange is the internal organelles, and we're looking through the ortho slice of the raw 3D volume of FIB-SEM. Now you immediately notice the difference of peripheral and getting close to the nucleus. You have very sparse organelles towards the peripheral, but getting a lot more dense when it gets to the nucleus. That's why it's very important to have large volume that contains the whole cell, so you know the statistic, you know these distribution of organelles. And here actually we have 2 cells that provide a certain amount of a statistic of cell-to-cell variation. Now let's switch over to the optical channel. In this case, we fluorescence label ER in green and the mitochondria in pink. Again, you see this anisotropic distribution of organelles, much denser towards the nucleus and much sparse towards the peripheral. Let's zoom in to examine the alignment quality. We use ER and mito as landmarks. They look pretty good. Now, this is a very interesting finding. It's a small vesicle inside a nucleus. So what is it? It turns out it actually contains ER proteins, which is a good surprise. Actually, as a matter of fact, almost every single data set that we have looked at, they are full of these surprising discoveries. So to summarize, let me just come back to this resolution volume space. To image a 3D volume using electron microscopy, there are a variety of complementary techniques that one can choose depending on the questions. So I've shown you the development of enhanced FIB-SEM that pushes the volume boundary of a conventional one, transforming FIB-SEM from a lab tool lacking long-term reliability to a robust imaging platform with 100% effective reliability, basically capable of months to years of continuous imaging without defects in the final image stack. The few examples that I've shown today, from the brain tissue to contains thousands of cells to whole cells in tissue, as well as high-resolution cell atlas. I think they're just the tip of the iceberg. It's the beginning, we'll say, of a holistic way of looking into the neuronal circuits and the cellular world, which would hopefully lead us to the better understanding of how the brain works and how cells function. So finally, let me end my talk with Sydney's quote again. I have shown you a couple new techniques, new discoveries, and propose some new ideas. I hope they will, if have not already, stimulate more new ideas. With that, I thank you very much for your attention, and I'm delighted to answer your questions.
What a wonderful talk there, actually, Shan. I'm amazed you can fly through the ER. It's just quite a stunning image, and it's also great to see how much work is going on away from connectomics, which was the original driver for this. Curti, are you ready to run
Yes.
an Mentimeter?
Yes,
Yep.
and here's the Mentimeter link again. I will just post it again so everyone can log to the
Okay.
quiz.
Yeah.
Until then, Shanzu, you can possibly look at the questions, some of the questions, and Nick will do the questions after the quiz.
Yep.
So what I will do is, yeah, the link to the quiz is just posted, and yeah, if you can log, and then I will just share my screen and do the quiz.
That hasn't quite shown up, but yeah, people, if you want to put some questions in the chat, feel free to just keep entering those as we go. Oh, here we are. Yep, got that. Thanks,
So
Kirsty.
everyone can see my screen, right?
Yep.
Awesome. So we will wait a few minutes. About 15-20 people is a good number. So yeah. So what's your order for progress in science? So since the talk started with Sydney Brenner's quote, so I thought like just play around techniques, discoveries, and new ideas. So you need new techniques, then discoveries, then new ideas, new techniques, and then new discoveries. Or often it happens, there's some random new discovery, and then that leads to a lot of technique development, and then leading to more ideas. So yeah, nice. I think this is we had 30 people after a long time participating in the quiz. So that's awesome. So yeah, just a couple of more seconds, and then we will start at the questions. And the questions are also very interesting. Um, so I think that's we have, and so 32 people. So this should be fun. And yeah, the faster you answer, the more points you will get. So what is the advantage of FIBSEM for volume EM imaging? Isotropic resolution less than 10 nanometers, fully automated, easy to register the image stack, all of the above. And Shanzu, if you want to comment on some questions, feel free to comment. So that's it. And the correct answer was all of the above. So the race comes down to 13 people, I guess. So we go to the next question. And oh yeah, we also have a leaderboard to keep things exciting. So PV, Shami are the top two ones, and others are also close. Magical Rhino, Dr. Centipede, that's a nice name. So yeah. The next question is, let's see. What is the typical duration that a commercial FIBSEM system can run reliably? 10 hours, 100 hours, 1,000 hours, 10,000 hours. I guess 10 would be too less. 1,000 hours will be too much. 10,000 would be remarkable, right? So, yeah, so 100 is a good answer, even if you didn't watch the talk. So let's see who is on the leaderboard, who is leading. So Lucy C has taken up. I hope this is not Lucy Collins. She definitely has an advantage. Oh, David is fastest actually. Anirudh and GP, MC, Maverick, everyone is closing up. So uh, let's look at the third question. What is the isotropic voxel resolution of the largest connectome to date? The Drosophila hemibrain. 4 nanometers, 6, 8, and 10 nanometers. So the lucky 13 people. So let's see how the leaderboard is going on. So it's a very close fight between Lucy and David. So I'm pretty sure Lucy is going to win now, just because of the name. Uh, let's see the last. I think this is the last or the second. Yeah, this is the last question, and a bit tricky one. So you should be listening to the talk to answer this. Which of the following is correct? Microtubules are longer in mouse pancreatic beta cells under high glucose. Endoplasmic reticulum primarily forms sheet structures in lean tissues. Hemibrain data was acquired on a FIB-SEM system in 2 years. None of the above. Yeah, 9 people were listening. So let's see who finally won. I'm betting on Lucy. So let's see. Oh, that was interesting, GP. I think this is for just this answer. Overall, I think Lucy has won. So I have configured it wrongly, because if you see overall points, 2701, and I think this last question, GP had a lead. I'm not sure. I have to check. But it's up to Nick to decide who's the winner, because Lucy shows on the top, and overall point I think are less. So possibly Lucy has won 27-0.
I have to say, well done, GP, as well.
Yeah.
I wasn't following the last round, so I didn't see. Anyway, that was great fun. There's some great questions there. We've got a few more minutes left. Should we just move on?
Get in touch with Georgina or Stephanie, yeah, for the prize. Yeah.
Yep. Oh yeah. Okay, so let's get into the chat. Sorry. All right. I'll start up at the top here. Okay. So we've got a few questions here. Thank you very much. From Paul. Do you want to? Are you still there? And open up. Here we are.
Yes, I am still here. Great talk, Shan. And I only got the first one right, but I got it the fastest. In your new design FIBSEM, you have the FIB beam parallel or horizontal to the beam. How do you move that beam, or is the block moving? And how can you keep that stable for so long?
A good question. So actually, we don't move anything. That's the beauty of this whole approach, because any diamond knife approach, you have mechanical movement. So this one, there's no mechanical movement. So what it does is, as the FIB milling the block will shrink. So the distance, actually the working distance, is slowly increasing, say at 8 nanometer a time as we go. So, and actually I use that this property to construct this inline focusing routine, so that I know how fast it moves. And then I say, okay, is this in focus or not focus? We don't necessarily have to be a perfect 100% perfect perpendicular. Even though it has a small tilted angle, the beam, the aberration is not big enough that we need to worry if we only image smaller areas. Now, if you want to image larger areas, again, I don't use any stage movement. Instead, I tile large movement into smaller, say 50 by 50 microns. And because I have that autofocus routine, I can optimize each individual tile separately, all without any overhead, the same mechanism during process. So each tile actually has its own focus stake alignment parameters. So I can maintain a uniform focus index, what I call, across a bigger area. But we don't necessarily have to be perfectly perpendicular.
Okay, yep.
Right, thank you very much for that. Oh, that was very clear. So I've got Penhang Lu. Are you there? Sorry, do you want to say your question yourself?
Sure, thank you.
Well,
so my question is, did you use a multi-beam SDM for those high throughput imaging?
Yes, that's a very good question. Indeed, we have a different approach right now headed by Ken Hayworth. So he and his team integrate the focus, the multi-beam. But you think about the multi-beam, it's not quite compatible with this FIB milling approach. So instead of having this FIB gun coming at a glancing angle, removing small amount of material, he has they call a gas cluster beam coming at say 20 to 30 degree angle, kind of showering from the top to remove a small amount of material and then image this much bigger, literally could up to millimeter size of area using the Multi-SEM. That's our next generation platform to hopefully finish the entire flight. Instead of the 8 machine for 1 year, you can use 1 machine less than 6 months to then get the entire flight finished. That is the different platform Hess Lab has been working on.
Right. There's a minor question from me. Why you put a FEI FIB on a Zeiss SEM, but not full FEI or full Zeiss platform?
Really good question, I have to say. This is all historical reasons. Back in 2010, when I first started this, Zeiss didn't have, they have they use a Seiko column they call Envision. But other than that, they don't have a good FIB column. FEI is very good at making FIB columns, but their SCM doesn't have as good as a resolution. So we try to pick the best out of the two back then. So we use the best SCM from Zeiss and try to get the best fit from FEI, so integrate them. So no problem for me to do this type of integration. Actually, I have a lot of fun. But the reason that these 8 machines you see behind me, they are actually, Zeiss came up with a new FIB design a few years ago called Capella. So right now we have full Zeiss machines. I don't call them FIBs anymore. I call them JICE. J stand for Janelia. So I'm not sure if the resolution is good enough. You can see this J6, JICE 6.
Yeah.
Okay, thanks very much. Actually, so Nikos Christakis had a sort of similar question to this one.
Yeah, I was just wondering, why did Zeiss pick the original configuration for the stage and the beam, and is there any disadvantage to your configuration? I guess not, but I thought I'd ask.
Yes, thank you for the question. Yes, not only Zeiss, all the commercially available FIBSEM from historical reasons, because FIBSEM was not developed for this type of application that I was just sharing with you, right? So the FIBSEM was developed a couple of decades ago, targeting for semiconductor industry. They're doing these, for example, failure analysis on the IC chip. So what they want is a very convenient to look at the same area, same location, both from the SCM column and the FIB column, so that they can locate the potential defect area from SCM, and then they fit into that particular area and see the cross section. So having these two columns at an angle provides great convenience to do such an approach. Now, same thing. If today, because our application are quite straightforward, we only need to, like the fly brain, right? We know this is a big area, we need to image the entire volume. But oftentimes, there are applications, if you have a culture cell on the cover slip, and you're not quite sure which cell you want to go to, or maybe you want to go to multiple cell in somewhat of a easier fashion. This 60 or 50, 60 degree configuration is still very helpful because you can navigate easily from the FCM, go to the place you want to mill, and then you mill that area. But the problem of doing that is you end up digging a trench into the volume there, and the trench is accumulating. Think about the gallium beam, and as well as the sputter material. It will cause certain amount of instability for the entire process. But that is the convenient or the pros and cons of these two different configurations.
Thank you very much. Thanks.
Thank you very much, Nikos. More questions coming in. I'm not sure. Well, I'm just sort of ask one. So the neural network part of this, the actual trained segmentation, is really a critical part of the whole process these days. Otherwise, it just takes you too long to look at the output. But I have a very naive question here. Let's say you have a trained network can I have that one too? Can I download that from you? Is this something that's portable, transportable between users?
That's a very great, it's right on. I think your question about the challenge of this whole volume EM pipeline or approach to study biology questions, because generating data, looking back, it's not the most difficult part. It is the analysis. So the pipeline that or the model that we generate, for example, what Google used to segment out the fly brain. There are two components. I'm not the AI expert, but I kind of know there are two components. One is the architect, the overall skeleton of the network.
Mm-hmm.
The second are these fine-tuned parameters, and the parameters basically to obtain these fine-tuned parameters, you use a lot of training data to do that. And the software code is all open. You can download it, but then you just need to train up your own network. You can use what existing, for example, for these flies, you can use whatever very similar sample as a starting point. You'll give you one prediction, then you go in and correct those manually. It's already much better than starting from scratch. Then you just go through the iteration and feed a better ground truth back to the model, train it again, and then see what else is left. And that would pretty much how we have been working. We always start with something existing, even though say now we have these this whole central nerve system, the CNS volume sending to Google. They will start with the hemibrain parameters as a starting point.
Okay, great. Thanks very much for that. That's quite illuminating there. I think we're both running out of time and out of questions. So let's just thank Shan for such a fantastic talk. I mean, this is really an interesting area that's just going to grow and grow and grow. I'd like to thank you so much for that. Everyone can do their awkward Zoom clapping or whatever. Oh, you're getting the hands, you're getting the claps there. There you are. Thanks very much from everybody. And also, I just wanted to mention that we got Rainier Kaufman is actually doing the talk on the 12th of the 7th on cryo super resolution fluorescence microscopy for correlation with cryo-EM. So sort of another extension of this general theme next time. Anyway, so unless anyone else got any more questions before we finally wrap up? No, I'm not seeing anything. No one put their hand up. Okay, well, in that case, thank you very much then, Shan, and we'll shut down for the day.